Okay. As the first speaker of the second half of the last session today, I want to introduce Ms. Linda Arhin from the Putra University in Malaysia. She is a PhD student and also IPPN Travel Award winner for this year. Welcome, Linda Arhin.
Hello, my name is Linda Arhin, PhD candidate at the University Putra, Malaysia. And thank you to the conference organizers for giving me the opportunity to come and present part of my PhD project on the topic, Phenotypic Variability Analysis of Dwarf Coconut Varieties for Yield and Yield Component Traits. How? Philip?
The big three.
Oh, okay. So just in case you're thinking of coming to Malaysia for your next summer holidays, I think you could look up some interesting places like the Batu Caves, the Batu Caves, the Langkawi, Genting Highlands, the Sunway Pyramid Lagoon, and also I must say that the Petronas Twin Towers is synonymous with Malaysia. Now back to coconut. The coconut is a perennial tropical crop, mostly found on the coastlines and areas with good rainfall distribution in tropical countries. And they are known for their hard outer shell and fibrous husk. They have both food and non-food uses, but they are normally cultivated for their water, milk, copra, and oil. Two main types of coconut, the dwarf and the tall. The dwarf grows to a maximum height of about 10 meters, and whilst the tall can grow up to about 30 meters in height. The dwarf are mainly self-pollinating, so they have a lot of uniformity in fruit characteristics within population, whilst the tall types are outcrossing, so they have a lot of heterogeneity within population. Some bit of information about the global coconut industry. 90 countries cultivate coconuts in the world, and the production covers around 12 million hectares of land, and majority of this is in the Asia Pacific. In 2022-2023, production was around 67 billion nuts, which translated to about 7.73 billion US dollars to global coconut smallholders. In Malaysia, coconut is the number 4 in terms of head trade after the oil palm, rubber, and paddy. And the major exporting coconut products are the coconut oil, activated carbon, desiccated coconut. They also import a lot of fresh coconut and coconut oil from neighboring countries like Indonesia and the Philippines. In the world, Malaysia ranks 5th in terms of productivity with about 7,106 nuts per hectare. 4 main varieties are recommended by the Department of Agriculture in Malaysia. The first one being the Matak, which is a hybrid, a cross between the dwarf coconut and the tall coconut, and the other 3 are all dwarf varieties. There are many challenges associated with the coconut industry in Malaysia, but the major ones are that there's inadequate high-yielding quality planting materials to supply to farmers, and there's also low domestic supply of coconuts in the country, and they also have a lot of unproductive old coconut plantations. In view of this, the government has come out with policies to help curb some of these challenges that the coconut industry faces. And one of them is the National Agri-Food Policy, in which the government has made it a priority to revive the coconut industry. There's also the RMK 11th and 12th Malaysian Plan, in which the government says that it's going to add coconut as a source of wealth creation in the country. To walk the talk by the government has provided funding under the Malaysian higher education under the long-term research grant for new hybrids with good yields or high yields to be developed to the existing Masa hybrid. And we know that to kickstart our hybrid development, we need to determine if there is sufficient genetic variation in the existing germplasm that we have. So we set the objective, 4 main objectives to achieve this target. And the first one, we want to explore genetic variability among the 90 palms. That is the 3 recommended dwarf varieties by the Department of Agriculture. And the yellow dwarf, red dwarf, pandan, so 30 of each variety based on their phenotypic performances. And we also want to know that if the variability that we see in the population is mostly caused by genetic factors or mainly due to environmental factors. And the ultimate aim is to identify some high-yielding palms that we can use to develop potential coconut hybrids. To start the study, we sampled some coconut palms in 2 places in the northern states called Perak, one of the northern states in Malaysia called Perak. And we sampled palms that were 10 years and over, because mostly with coconuts, when they are 10 years and over, that is when we have stable yield. So we sampled 30 palms of each variety, and we took data on the fruit yield, the palm height, the girth, and all other parameters. To give you an idea of what we did with our data collection in the field, this is mainly what we started with. We started with the average nut yield per palm per year. So we go to the field, we look at the 3 most matured branches on a palm. We count the number of fruits in that, and we calculate the averages. In our next count, we do same thing, and at the end of the day, we estimate the average number of fruits per bunch to get how much that particular palm, the yield of that particular palm that we've selected. And for the palm height, we measured the stems in a straight line from the ground to the base directly under the crown of the palm. And we did this using, for palms that were 3 meters and below, we used a 3-in-1 laser distance measuring device. But for palms that were 3 meters and above to about 5 meters or 6 meters, we used the UAV to help us in doing this. For other features or traits that we took, they were mainly on the flowers and on the stem, and these were done by hand using simple tape measure. So after analysis, we for the ANOVA table, we realized that there were significant differences in the genotypes for all the traits that we measured. And this indicates that there's sufficient genetic variability in the genotypes that we were studying or in the palms that we were studying for our hybrid development. Also, for the studies on the genetic parameters, we realized that between the genotypic coefficients of variation and the phenotypic coefficients of variation, there were marginal differences. This also implies that the expression of the traits that we were measuring were less influenced by the environment. So that means that we could choose those traits based on their phenotypic characters. For the heritability, we had high heritability values, also indicating less influence of environment on the expression of the trait, and that also the traits could be passed on to their next progenies. From cluster analysis, we had 5 main distinct groups for the 90 palms that we studied, except for cluster 2, which some of the varieties grouped together, or the other clusters grouped into their distinct varietal groups. And also for the PCA, we realized that principal component 1 and principal component 2 gave a variability value of about 63%, and the traits that were giving most of the variability were the number of fruits per palm, the palm height, they gave at 20 centimeters, and gave at 150 centimeters. We did the correlations, and we realized that the palm height, they gave at 20 centimeters, they gave at 150 centimeters, and the number of racula were strongly correlated to yield, so meaning that we could look at those traits and select for yield. So in conclusion, I'll say that yes, there was some variability in the population, the 3 populations that the DOA recommended varieties, and most of the variability were due to genetic influences than the environmental influences. And the correlation and PC analysis also indicated that we could select high-yielding palms based on some traits like the palm height, the racula, and the girth at 20 centimeters and 150 centimeters. So all in all, we were able to select about 12 palms, 4 of each variety, to proceed a hybrid development project. Thank you to the organizers of this conference once again for the travel grants to be able to share my work with you. I also like to thank the Ministry of Higher Education Malaysia under the long-term research grants for funding the research, and to my wonderful supervisory committee. The last not the least, I'll say thank you to Mr. Afolabi of A&M Texas University for the final touch that he made to my presentation slides. As Malaysians will say, terima kasih. Thank you for listening.
Linda, thank you for speaking. So we have enough time for a few questions. So I would like to open the discussion. Maybe I'll start with one question from my side. So since palm trees have to become quite old before they can be harvested and yields can be collected, and you said that height was a very important determinant of the overall yield. So could you already now project some recommendations for management based on the results you obtained during your PhD? So for example, what palm producers or coconut producers could do better to increase their yield based on your results?
Yeah, so basically when you go to the field based on my results, you could determine from maybe the palm height, how tall the palm is, or how big the bowl or the trunk is, to know that if this coconut is going to yield higher or not.
Can producers and farmers do something to increase the height? Is there a go-to way, for example, the removal of understory while the palm tree is still young or something like this? I have honestly no clue about palm trees.
Yeah, so I think most of these traits are more genetic than environmental. The height and the width of the stem, because even with coconut, it lacks the cambium, so when it expands to some size, it cannot expand again. So most of these traits are more genetic than environmental influences. So I don't think farmers can do much to increase the height or the weight of the palm.
Except selecting the right trees.
Except selecting the right. Yeah.
So pre-breeding, or even this is a job for gene banks to pre-breed the best.
Yeah, the best palms before giving to farmers. Yeah.
Yeah. Okay. Yeah. Well, I mean, that is definitely a take-home message here for the gene bank managers. Any other questions?
How long does it take for a coconut tree to produce coconuts from germination to the first coconut they see?
If I'm getting your question right, after harvesting the coconut fruit, how long does it take to sprout and develop into a stem? Is that what you're asking?
From the sprout to a full tree and having another coconut.
Okay, so let me start from the pollination. When coconut flowers are pollinated, it takes approximately 11 to 12 months for you to have the full fruit, for it to dry up, harvest the seed, go and put it in the nursery, and you have to wait for another year for it to sprout to some particular level before you transplant to the field. And for dwarf varieties, it takes about 3 to 4 years before you can have the full coconut and have some fruit. But for the tall variety, you have to wait for about 7 to 9 years.
Long process. Thank you.
So it's more comparable to forestry here than to a crop, than to a field crop. Any further questions? No? Then I want to thank Linda one more time, and thank you very much. And we'll go over to our next speaker, one of the long-standing members of IPPN. It is my special pleasure to announce the Scientific Director of the Plant Accelerator from Adelaide, Australia, Dr. Bettina Berger.
Thank you very much, Philip. I guess I take it as a compliment rather than making me feel old. So I know it's Thursday. It's day 4 of the conference for most of us. It's probably beer o'clock somewhere already. So we'll take things a bit slower. I'm mostly going to tell you a story. There will be results in my presentation, but they're really not relevant to the story. So really just feel free to relax, and mostly I'll summarize the key take-home messages at the end if you miss things in between. So Richard already made a really good intro of the APPN yesterday, so I don't have to talk about that much. So just want to mention that within the OzBarley project, most of the work was done initially at the Adelaide node. We now also have work at Western Australia and in Wagga Wagga as part of the project. And we're covering everything from the controlled environment to field phenotyping with this project. So barley is a key cereal crop in Australia. It's the second largest crop after wheat. And about 30 to 40% of the world barley trade is Australian malting barley. So tonight, if you're drinking a American beer, that's likely going to be American or Canadian barley. If you're drinking a Mexican beer, there might be a chance that that contains some Australian barley. But really our biggest importer or trading partner for barley is China, and also the Middle East, mostly for obviously feed barley. So barley production in Australia faces a number of challenges. We did have recently some trade disputes where heavy import taxes were imposed, which heavily impacted the industry. They have now been lifted, but we do obviously also have ongoing environmental fluctuations, and they are not getting any better with climate change. And then growers obviously face the constant issue of heavily fluctuating prices, which can massively impact how they can market their grain that they produce. And also further downstream for the brewing industry and maltsters, they are a very electricity and water heavy industry. So there's obviously also the challenge of reducing the environmental footprint there. So we've heard a little bit during the conference about various germplasm collections for sorghum, for example. Harkemal mentioned one of the rice diversity panels. And we got together as a group and discussed the lack of a barley germplasm collection that sort of represents what's going on in Australia over the years, and also something that could be used by the industry. So we got together a large team, and I'm putting this slide here at the beginning so I can thank everyone involved rather than potentially forgetting something at the end. So we did have an opportunity to get funding to get this project off the ground. Interestingly, that came partially from the ARDC, which is the Australian Research Data Commons. So you wouldn't necessarily think that a facility that's mostly focused on creating data infrastructure would invest in a barley project, but it was really about generating a data asset that meets the FAIR standards that could be used by the community. And then APPN obviously co-invested. Bioplatforms Australia is our sister facility, so they heavily focus on the genomics, proteomics, and metabolomics side. And more recently, the Grains Research and Development Corporation invested in MEL. We also had a number of public-funded research institutions mostly contribute their time and goodwill. Importantly, wrong button. Most importantly, the Australian Grains Gene Bank was involved right from the start. And also we had all of the 3 major commercial barley breeders in Australia on board, and they contributed heavily in terms of their in-kind support. So how did we go about creating the OzBarley panel? We pretty much put out a call to the community. We talked to the breeders, to the main research groups around Australia to come up with a wish list of all the barley lines or cultivars that had been released over the decades of barley breeding in Australia that should be included. We then sourced the material primarily from the gene bank, but also some from breeders and other contributors. Peter mentioned that material in the gene bank can sometimes be heterogeneous, which is perfectly normal. So we went through 2 rounds of single seed descent to create genetically pure material, and then bulked up the material in the greenhouse. In parallel, we also did, mostly Julian, did an awful lot of detective work trying to piece together this very complicated, pretty much pedigree of Australian barley breeding. I don't expect you to be able to read anything here, but just to show you how complex pedigrees can be in breeding programs. And the ones you can see at the bottom, they are lines that are currently grown in Australia. What was also important when we put together the panel is that there were no IP restrictions on any of the lines that we were using, so that the material could be easily shared later on. So just zooming in to give you a bit of an idea, there are some lines like Proctor here or Clipper that are highly important because they are the parent of many lines within the pedigree. And then there are other lines that, that are pretty much once off in a sense that they were bred but didn't really feed into any of the further breeding programs. Okay, so we've got the material, we've got the pedigree. We obviously wanted to genotype the material. So in addition to using an Affymetrix SNP chip, we also used RNA-seq as an alternative method for sequencing. We didn't have the budget to many different tissues and organs, so we really focused on germinating seedlings because that has obviously relevance for the brewing industry as well. And that was all done by our colleagues at CSIRO and the AGRF, which is the sequencing facility in Australia. So for our SNP chip, we've got about 12,500 SNPs, which is sufficient to give you a pretty good coverage across the barley genome, but it's not really high density. So with the RNA-seq, we could then enrich that to over 100,000 SNPs. And you can see here, for individual genes, we have quite a number of SNPs. Obviously, RNA-seq is limited to transcripts that are present. So this gives you an idea of what the results may look like. So at the very bottom, we have the MOREX3 reference genome. We also have MOREX in our collection, and thankfully there is not a single SNP in that region, as you can see. It's always nice when your version of MOREX turns out to be the same as the reference genome, which isn't necessarily a given. Similarly, CM72, which is another 6-row barley, again, no SNP in this specific region, whereas Wimmera, you can hopefully see those vertical lines. Each of those represent a SNP that is present here. But then we also have the case where 2 of the varieties don't have the gene expressed at all. So we do get quite rich information from this type of genotyping. So we have the pedigree, we have the genotypic information. How do they compare? And obviously with genotypic information, the classical approach would be to do a phylogenetic tree. We tried something slightly different just because of the way the relatedness was between the lines. So we created a net that in a sense shows you the distance of different genotypic difference between the lines. You can see here, that's mostly where some of the European ancestor lines cluster. Down here, we have some of the North American ancestor lines. Over here, some of the 6-row Asian lines. And then in this cluster is a lot of the modern Australian cultivars. In addition to the land races, where we've got about 220 lines, we then also to the elites, we then also added land races to the story. And we thought if we do a PCA on the genotypic data that we would get nice separate clustering of the two. That wasn't the case. So you can see that the land races and the elites cluster together for large areas. But we do have some clusters that are, in a sense, landrace only. We were trying to select landraces that have a broad geographic spread and are sort of semi-dwarf. They weren't all that dwarf when we grew them in the greenhouse, so they obviously come with big challenges. As Peter already mentioned, dealing with this type of material is not like dealing with all the elite ones. So we've got the genotypic data, we've got the pedigree, we've got the pure seeds. So we then proceeded with phenotyping, and let me see if this works. Yep. So we did sort of a baseline phenotypic screen in our controlled environment facility where we literally just wanted to look at growth over time. We also harvested spikes at the end, put them through our X-ray scanner, so we can look at spike architecture and seed morphology. And you can see some lines that are a lot earlier flowering, like unicorn, for example, that's represented in the growth curve peaking earlier, versus some of the sort of later developing lines like dictator further down. Okay, so we've got genotypic data, phenotypic data. We can obviously do GWAS. This is really just a simple one for looking at row type. And I'm not going to go into any of the loci or anything. That's really just an illustration that we can do that. With the data we've got, this is work from one of my PhD students. He's interested in root growth angle. Now, in most places around the world, you are looking for deep roots with a fairly shallow angle. South Australia, we don't have great soils, so mostly what's down there is toxic and not much water, so we're actually looking for a shallow root system. So more like the one on this side here. And again, we can use the phenotypic data, combine it with the genotypic data to then do GWAS. Okay, so how do we put it all together? We've got genotypic data, pedigree data. We've got some basic growth analysis, some basic phenotypic traits like height and other aspects. For the elite panel, more basic phenotypic for the land races. And then we were looking for a database where we could upload all this data and make it available. And that's where we then realized that such a magic database does not exist. So we've used a bit of a hybrid approach. And when I say a bit of hybrid, you can see that it's quite fairly hybrid. So we have a landing page on Figshare for the sole purpose that allows us to mint a DOI, which we obviously want to use for tracking usage of the resource. We are using Germinate, which is a software developed by the James Hutton Institute that can host the genotypic data, the pedigree data, some very basic phenotypic data as well. The large numbers of image data is too big for Germinate to handle at this point in time. So the raw imaging data is hosted at PAWSI, which is a data storage facility funded by the national government in Australia, similar to APPN. So that's where the raw image data is hosted, and there are hyperlinks from Germinate to the respective data entry at PAWSI. The RNA-seq data as well, the raw data, really not suitable for Germinate at this point. The SNP data extracted from it will go on to Germinate, but the raw data is on SRA. So that gives you the infrastructure. Now, what does Germinate look like? We haven't published the paper yet, but if you know how to use Google, you'll find it. So this is pretty much the landing page. Gives you information about the germplasm, the markers, the traits, and location of the material that is in there. You have a menu over here where you can apply different filters and search terms and so on. So here's an example, and I don't expect you to read any of this, but I just filtered for a trait, which is peduncle length. So the peduncle is the bit of the stem that is pretty much this bit here between the last node and the spike. And you then get a box plot for the entire data set. And you can even click on individual dots, and it then pulls out which variety that is, or you can download the data in CSV format from Germinate. So if we then click on one of those examples, so we then get the passport information for Germinate. We do have a pretty good passport completeness index. We had a very dedicated research officer spending a lot of time trying to get this passport score as high as possible, and we'll soon add DOIs when we deposit them back into the gene bank, so that'll lift it even a bit further. You can also have a look at the X-ray images, so projection of the individual seeds, or a slice through the spike for each of those entries. And we then also have a link to the raw images. So these are hyperlinks where you can then click on and get the raw images that were collected during the growth analysis. So this is all controlled environment phenotyping. Obviously, barley isn't grown in a greenhouse for production. So we also wanted to look at traits and phenotyping out in the field. And we took a bit of a different approach for this, which is what I'm going to call community-based field phenotyping. So we had funding from the GRDC to run trials. So we've got 3 trials out at the moment in Western Australia, South Australia, and New South Wales. And we'll have another set of 3 trials out next year. And what we did is we put out an open call to researchers across Australia, everyone from PhD student, early career researchers to senior academics, to say, we are going to run these trials. If there's a specific trait that you want to phenotype, you can come and access the trial, as long as it obviously doesn't destroy the trial. We also had some travel support for students and EMCRs. So what we did, put out this call, we got applications in, and there were quite a number of different traits that people were interested in. Some wanted to look at spike architecture. Some wanted to look at head loss, which is a big agronomic problem in Australia, where entire heads snap off, fall to the ground, and are obviously lost to the farmer. Lodging can be a problem. We have salinity widespread throughout Australia, so sodium and potassium accumulation can be an issue. We also have a PhD student who wants to look at things like nutrient use efficiency. So the photos you can see here are pretty much from this year, where researchers went out to do measurements. Some accessed just one of the trials. Some were accessing all 3 trials across the country. We will also have yield data from 2 of the trials. South Australia, unfortunately, didn't get any rain during the critical period, so that one will probably turn into chicken feed, which is rather sad. But yeah, what you can see here is us sampling leaves for salinity tolerance measurements. So we're looking at sodium accumulation, very cheap trait to measure. And these are traits where researchers look at photosynthetic traits at the trial in New South Wales. And what that really allowed us also was to connect researchers with each other. So we had applications coming in from someone in Western Australia and in New South Wales, both wanting to look at photosynthetic traits. So we were then able to connect them up and say, hey, you're wanting to measure the same thing. A, you can probably help each other out so you don't double up on work, but B, likely to have good opportunity for future collaborations. So with this, I just want to summarize that I believe that working across the omics areas can create a resource for common use. And it can also reduce the barrier of entry especially for early career researchers or researchers who don't come from labs with massive funding. So as an example, this is Harley. He's an honors student. So honors students in Australia, they have about 9-10 months for their project. So there was no way he could create or collect a germplasm resource, get the genotyping done and phenotyping, because of the time, but also because of the cost involved. But by having those field trials out there and all the data publicly available, he can pretty much go out to the field and simply collect the leaves, do the measurements back in the lab, and do the GWAS analysis later on that data. So I think investing in a national data asset like this can also create new collaborations. As I mentioned, we were able to connect researchers who were working on similar subject areas that didn't necessarily know of each other's work. And with that, I just want to say thank you to our funders and to you for your attention. And I'm going to leave you with a photo of the OzBarley trial in Western Australia, which was taken about 2 days ago. Thank you.
Thank you very much, Bettina. And the room is open to questions. Rick?
I think there's a prize for asking the most questions. I'm on it. Now I was thinking about, I'm going to ask a silly question here, but the roots, right? You were selecting the phenotypes that have shallow roots because of the soil that is toxic. Apparently your soil is only 30, 50 centimeters, I once learned. Aren't the roots smart enough that if they hit the toxic zone that they simply decide to go shallow?
They might, they will likely divert eventually, but there is one aspect which is growing into toxic soil. The other aspect is, in a sense, wasting resources by potentially growing into soil where there is no water. South Australia has a so-called Mediterranean climate, so we have intermittent rainfall. So the shallow root system actually helps to capture that intermittent rain that comes during the season. So having that shallow root system right from the beginning will help plants to then capture that precious bit of rain that comes most years, not this year in South Australia, but thankfully it did come in the other states.
Okay. Thanks.
Mm-hmm. Yeah, very nice talk. I'm interested in this community approach. I think that's a very good idea. But are there any, did you select people, or could everyone join for every type of research, or how did you organize that?
So I guess the restrictions we had was the trial had to remain intact, so we couldn't allow someone to do, biomass cuts halfway through the project, because that obviously would then ruin the trial for others who want to do harvest traits, for example. The other aspect is that certainly for the ones who receive travel funding, there is a condition that after a quarantine period where they can work on their data to publish, that data then becomes publicly available under CC BY4 license, like everything else in the OzBarley project, and then becomes part of the Germinate portal as well. So really the idea is that we funded the creation of the resource. We had funding to run the trials, and people can access it, but the expectation is that they share the data to then grow the overall data asset for others to benefit as well.
And were there any things that were not studied that you hoped that people would come for analysis, or did you have the full spectrum of things that you want to acquire?
I think there are always more things that you would like to measure. So we probably didn't have much metabolomics or things at this point. We will likely have someone looking at proteomics of the mature grain at harvest to look at protein content. Some people wanted to look at disease tolerance. That was one thing we couldn't facilitate because these trials were managed according to best agronomic practice, so we couldn't run a disease nursery. But yeah, that is where the limitations are.
In the back.
Thank you, Bettina, for sharing this nice resource. I just had a question on the data set. So the RNA-seq data for this panel, is it sparse, that it's only good for SNPs, or you could actually do functional analysis at expression level?
We didn't have the resources or the financial resources to do multiple tissues, and we also couldn't do replication. So it's really mostly for, I guess, presence, absence of expression. And SNPs, it's not good enough to look at expression levels.
Thanks.
Great talk. Do you have consideration to integrate the Barley ontology into the data platform? There is a trait dictionary existing for barley.
So the traits that we measured, sort of the existing traits, things like, height or yield or sort of established phenotypic traits, we try to use ontologies as much as we possibly could throughout the project. There were some traits, obviously, that we measured with the phenotyping platform where we had to create our own vocabulary. Again, for this, we tried to use best practice. So we worked with people that work in the ontology and data vocabulary domain to have that as sort of consistent and standardized as possible.
So you have the ontology in the Germinate application. Is that what it is?
Yeah, so the traits that we measured, where there is an existing ontology term, we would use that ontology term, yeah.
Yes.
Okay, thank you very much, Bettina. One more round of applause, please, for her. Then I would like to call to the front all the flash poster presenters and get ready to present your posters, please.
Hello, everyone. My name is Masa Buden. I'm a PhD student from Novi Sad, Serbia. Also, I'm a junior researcher at Bioscience Institute. And today I'm going to give you a quick introduction to the experiments we did this summer with soybean seeds, where we experimented with different seed priming techniques. We used exposure to high temperature, to low temperature, to sulfuric acid, as well as the microwave treatment that you used as a control as a dead seed. Also, we had a control with zero treatment. We took pictures with thermal camera. First day, we took pictures every hour for 8 hours after treatment. And then every other day for 8 days, we took pictures once a day. And the results show significant difference between treatment with low temperature and the control. So if you have any comments, suggestions, questions, you can ask me, or you can see my poster number 89. Thank you very much.
My name is Alex Feldman. I'm from Kyushu University in Japan. I'm a researcher in Koji Noshita's group. He presented earlier in this session and kindly introduced my work. We quantified the three-dimensional shape of 109 potato tubers using the spherical harmonics in order to summarize the morphological diversity of 11 cultivars in an empirical morphospace. We did this because potato shape traits are very important for genetic studies and breeding. However, current studies are mostly limited to one-dimensional and categorical measurements. This reduces the descriptive capacity of the measurement, and we therefore propose a practical workflow to generate continuous quantitative descriptors of 3D shape. I'll give an overview of the method. First, we used photogrammetry to generate high-quality 3D scans of each tuber. We did this with an automatic turntable and 4 cameras. We used Agisoft Metashape software to generate the 3D mesh polygon data. Next, we converted the mesh data to voxel data, which is required for spherical harmonic analysis. Our workflow can handle other types of 3D data, so you can bring your own mesh or voxel data and enter the pipeline at this point. Finally, we generated spherical harmonic coefficients of each specimen using the SPHarm PDM toolkit in the Slicer Salt software. Basically, spherical harmonic analysis extends the same concept of elliptic Fourier analysis to 3D by calculating coefficients of functions which approximate curves along the surface of the sphere using a point distribution model. We get over 2,000 coefficients per specimen, which can be used to reconstruct the shape quite accurately. Then we perform dimension reduction on the dataset using principal component analysis to find a latent space that describes the variation in the shape. We call this space an empirical morphospace because each axis describes an aspect of the shape variation. The figure on the right side shows the occupation of shape variation in the PC1, PC2 space. Each point is a specimen colored by the cultivar, and the shapes in the background are showing what the shape will look like at that location. We found that the first 4 PC axes describe biologically meaningful aspects of the shape variation. By the way, this can be applied to other structures like fruits or seeds. Everything is done in Python, and we'll share our code and dataset soon. Thank you.
Hello, my name is Ashlyn Rairdin, and I'm a PhD student with Dr. R.D. Singh. And this is an overview of my poster using multispectral UAV imagery and machine learning for yield prediction in mung beans. And so our objective was to build a pipeline to use UAV multispectral imagery in our breeding program. And then we wanted to take that pipeline and first characterize the adaptation of our advanced lines, and then also find what are important characteristics and time points for mung beans in Iowa. And so to do this, first we planted 15 lines in 4 locations across Iowa. And then UAV data was collected weekly starting 2 weeks after planting, about when emergence starts. And we used a MicaSense Red Edge dual camera system, which has 10 spectral bands, and several indices and canopy coverage were calculated for each time point. And so first, the graph on the left, this is preliminary data from 2023. We also did this in 2024. We wanted to look at correlation between some of these vegetative indices, canopy cover, and yield. And so some select indices are shown here. And what we want to see is where can we find a higher correlation earlier in the season so we can then use that to make selections earlier in our program. And then also on the graph on the right, we're looking at the comparison of 3 different lines, including a diverse accession, a commercial check, and our elite line across the season with the NDVI value. And what we see is our elite line actually has a higher NDVI value earlier in the season, which gives us hope moving forward with the yield prediction that we will be able to find a way to do some strong yield prediction with our lines in Iowa. If you want to find out more, my poster is number 85, or you can also come talk to me. Thank you.
Hi, folks. It's a pleasure to be here. I'm going to talk how can we use gene expression as molecular phenotypes to understand more of the genetic basics of expression. Okay, so we know that gene expression is associated with changes in final phenotypes that we can measure. And then there is at least 30% in maize gene that has been associated with a variance in the genome. So we can use these variants to fine-tune the expression of those genes. So commonly, when we are doing some analysis, we are trying to knock out the gene that is going to get the minimum expression of the gene, but there is a whole range of this expression that we can use in our favor. So to use that expression, we need to know which are the variants that are controlling those. So we need to be really careful in this step because this will depend if we are using the right gene or mutating the right variants. So we don't have a time machine. We need to be like the more precise, the better. And we are trying to answer this question: how stable are these variants? So for this, we are using two large gene expression data sets, one coming from 2020 and another for 2021 in maize. We are trying our best to remove all the external factors that are affecting these gene expression data sets. So we are using a set of genetic markers of around 15 million that has been already published. And we are finding these, dividing these regions that are controlling these molecular phenotypes in expression that can be in cis or in trans. So this is still an ongoing project, and there is like at this point no clear answer of how stable or how variant in the whole genome these regions are. But I want to talk about one example that is MATS1. So MATS1 is controlling flowering time. And if you see at the top panel of A, we are finding two peaks. One is in cis and the other one is in trans. So if we look at the cis that is in chromosome 9, we can see like, oh, maybe it's the same region. So we can knock out those variants. We can modify those variants, and we are going to modify the expression of the gene. But if we zoom in panel C and in panel D, we can see that those variants are different. So maybe this is depending on some other factors that we are trying to understand better. Maybe this is just how like this variation are like developmental stages, or maybe this is just like different things. So then we can open some question like how this regulation of the gene expression is in another species. Thank you. I'm happy to take any questions after these or comments.
Thanks to all the poster presenters, very nice. And now I would like to use the remaining 15 minutes to call back to the front Peter Wenzel to continue the discussion about gene bank phenotyping. And while Peter is sneaking up the stage, I already prepare one question. Peter, when looking back at the talks that we heard during this session, were there some aspects mentioned that you would consider of interest now in regards to gene banks, maybe taking up or putting a focus on coconuts or barley? What was the name again of that little thing on the barley between the ear? Pardon? Peduncle. Maybe peduncle length?
I learned a new word, yeah.
Yeah, me too.
Well, I think the barley example is basically what I would like to see us gene banks doing as we assemble a new collection. I would love to be able to have it everything like organized like you have organized now in terms of all the data arranged, curated, condensed into Germinate with some satellite databases linked to, and very high passport completeness index, which is for us impossible to achieve because we are sitting on historical data that you can check to a certain degree, but some things are simply missing and can never be recovered. So that's a huge resource, and it's a huge advantage in terms of targeting traits within the collection, which we don't have in most of the very large collections in the CG centers. And that's the reason why it's so important to characterize and evaluate those collections much more deeply. Because when I look at our bean collection, there are thousands of accessions which have a number and that's it, for which we sometimes even are not completely sure about what kind of adaptation, what kind of growth habit they may show. So we need to take them to to different sites. We have multiple sites of multiple elevations in the Andes to regenerate different, differently adapted bean accessions. And sometimes it's just trial and error because we just don't know almost anything about those accessions. So yeah, I mean, this is such a great resource. I was very impressed.
Peter, with the genotyping becoming more affordable and with a lot of the gene banks now routinely, running SNP data across entire collections, do you think it'll be easier to sort of put together those panels that represent a large proportion of the diversity that could, in a sense, then become a resource for the community so that, you could potentially, or I guess the community could search for funding to then grow those lines and invite people to come and phenotype for their trait of interest?
Yeah, I mean, your model of community-based phenotyping is fantastic. And we had a brief conversation, I think yesterday or the day before. It's a great model. We would love to do something similar in Colombia, but we don't have so many research groups in Colombia focused on beans, for example. So it would require international travel and everything, so it becomes more complex. Yes, essentially, yes. I mean, our strategy is to first genotype, ideally, the whole collection. We have finished cassava. We are lacking still resources to do the other two. I think we probably will have soon 10 to 12,000 accessions, bean accessions genotype, but there are 38,000. There's more required. So then initially, like putting a few tens of thousands of markers, ideally with a methodology that minimizes assortment bias to describe diversity and then condense into a subset, a core collection, and which probably should be whole genome sequence then and become a resource for the community. Erie for rice did something similar. They didn't genotype the whole 120,000 accessions. They selected a subset of 3,000 and whole genome sequenced them straight away. And as soon as those sequences became available, suddenly everyone wanted to phenotype those 3,000, which is, so it creates a community dynamics that leads to stacking of traits being characterized for those subsets. So I think that's definitely a way to go. And as you stack traits on the subset, if that subset is cleverly chosen, I think you can then use anything that can be learned from machine learning to extend and predict similar traits in the non-characterized but genotyped accessions as well. So to sort of spread the knowledge around as much as possible. Here I am. More questions.
Yeah, of course. Now, I've been visiting several gene banks worldwide. Japan, Bosnia, and of course, Belgium. Is there some trends that you mentioned this community interaction between gene banks to become more automated, become more data-driven? Did you see some developments past years?
Yeah, I mean, this is something I'm hoping for, waiting for, and trying to contribute to. Amongst the CGR gene banks over the last 1.5 decades, we were mostly occupied with getting our house in order, to be frank. Because we, like 1.5 decades ago, we came, or the various CGR gene banks came out of a period of chronic underfunding. And there was a huge backlog of accessions that required urgent regeneration. So really, during the last 15 years or so, we were just maximizing the numbers of accessions to be generated just to keep the collections alive. And we're sort of getting to the end of that tunnel, so to speak. And now I think there is clearly a movement towards now that we have secured the conservation, let's try to make it easier to use those collections by creating value-added data, right? Automation, to a degree, I think, automation, global leaders in automation gene banks are probably in Japan and China, Korea, not CG, not other national gene banks so much. No, that's right. So we have a lot to learn from them, I think, from those. I remember I haven't been there, but in Japan, the gene bank is everything. You don't go into the cold room and retrieve an accession. You push a few buttons on a screen, and the robot arms go in and take out what you need.
I was there. I made a video.
Are you were there? Okay.
I can show you later. It is fascinating, actually. Yeah. Another question, actually, we have a cool project that's about exploiting crop wild relatives in breeding programs. We've been running some trials with multiple crops, but the crop wild relatives are nasty plants to grow. They are wild. I would like to hear is that a common issue that you are struggling with, that they are difficult to phenotype, basically?
They are bloody difficult all the way around. Yes. And actually, I mean, in terms of conservation costs, it's probably each accession of a crop wild relative is at least 5 to 10 times as expensive to conserve as a cultivated counterpart usually. I mean, crop wild relatives are of great value if breeders run out of options, particularly disease resistance, for example, right? But they are very expensive and difficult to conserve, and even more difficult to characterize at the phenotypic level. Genotypically, it doesn't really matter. As soon as you can get out clean DNA, it doesn't matter. But phenotypically, yeah, it is a big headache.
Yeah.
And I mean, I've shown one of the photos of a guy walking in the screen house, surrounded by this huge canopy of climbing beans. These were wild. I think these were probably wild relatives because of the station where they are grown. And yeah, I mean, in the CG, we have to fulfill certain quality or KPI standards to qualify for ongoing funding from the Global Crop Diversity Trust. And one of these KPIs is that 90% of all of the accessions we are conserving are available for needed distribution globally, meaning that have been regenerated in sufficient numbers and have been tested, in our case, for about 40 different diseases each seed lot to ensure that they're clean and can be distributed. To get to those 90%, most of us were focusing on the lead material, or the primary gene pool, but not the secondary and tertiary gene pool, because to produce seeds of wild bean species, it takes 2 to 3 years for a good part of the species, whilst for Phaceros vulgaris in landrace takes you 3 months. So there's a huge difference.
Yeah. Okay. Yeah. Thanks.
You worry.
Any further questions? John?
Hi. I had a question about how you. I'm curious to know how you think about rapidly changing sequencing technology, and so the balance between storing sequence data now and how easy it's becoming, and like the advent of long read sequencing. I'd be curious to hear you just talking a bit more about that. Like, we know structural variants are important for traits, and up until recently, it was hard to get long reads for as cheaply as it is now. So,
no, we're not into long reads yet because we're not really going for resequencing whole genomes, etc. But actually, most of the CGI gene banks have done is to settle on one methodology, so we can also compare collections. For example, we are conserving cassava. IAT is also conserving cassava. We use the single methodology to genotype and compare both collections. In terms of structural variants, what we found was that we're using the DART-seq technology, which produces, essentially it's a reduced representation sequencing approach that produces both SNP data and also presence-absent variation data, essentially. And we always find, as soon as we step out of a single species and compare multiple species, these structural variants, of course, become very important for distinguishing for estimating genetic distances among accessions. But again, few of us have reached the point where we're starting to think about long reads and whole genome resequencing of large proportions of our collections, except Erie, who have whole genome sequenced several years ago already, 3,000 rice genomes. But they have the advantage of dealing with a relatively small genome. I mean, our colleagues in CIMMYT have a much bigger headache with their wheat collection of decent size. You have a follow-up question?
No, but that was really interesting. But yeah, I guess I've heard someone say to me once that the best way to store DNA is in a cell that you keep intact. And so in that sense, like if you can keep, like, you've got the seeds, is it worth? The seeds are being stored and that's pretty permanent. It seems like they could be really good resources just for, as questions come up, you've got the seeds and you can use the best sequencing technology of the time.
Yeah. I mean, so storing seeds versus I think that they essentially, several years ago, we were dealing with RFLPs and RAPIDs and AFLPs. Those platforms are not really future-proof or forward-compatible. As soon as you have at least sequence tags, you will be able eventually to integrate that into whole genome sequences as they become available. So in that sense, I think as long as you're sticking to sequence-based genotyping technologies, which everyone is doing today, essentially, I think you have a degree of future-proofness built into the system, fortunately. I was going to mention something else, but just skip.
That's okay, Peter. The time is up, and I would like...
The beer is ready?
Yeah, the buses are getting ready, and I think you should do too in order to attend the conference dinner. I would like to thank Peter one more time. Thank you for being here.
Thanks for the invitation.